So hello everyone, welcome to the week 11 of this NPTEL immunology problem solving session. So this week 11 we will discuss about the autoimmunity. So initially we have just to revisit, we discuss about the B cell activation, maturation, differentiation, T cell activation, maturation, differentiation, about antibodies, about some tools and techniques, complement systems. So after that we will first start with the autoimmunity. What is autoimmunity? According to you, what do you call autoimmunity? What does it mean? Anyone? Autoimmunity. So autoimmunity is when your own immune system is targeting your own cells, right? So when your self antigen is being attacked by your immune cells, then it will be called as autoimmunity. The simple definition of what you can think of. So if you remember during T cell maturation, because all of these things, all of these immune actions, especially for the adaptive immune response, the one of the major key players is T cell, right? T cell can activate the helper T cell. When they gets activated, they can activate the cytotoxic T cell, they can activate the B cell, they can activate the macrophage. Okay, so now after this activation, when they can activate this such a big pool of immune cells, then for example when it will activate the macrophage, then it will do the phagocytosis, it will clear the incoming threat or the antigen, right? When it will activate the B cell will produce antibody, and then antibody will bind and neutralize the antigen, right? And it can also initiate the complement action, which will lyse the cells, lyse the target cell, and it will induce the clearance of this antigen antibody complex, right? And also it can activate the cytotoxic T cell, which will show the cytotoxic activity against its target cell, right? So that's how T cell is a very major player here, because it can control the whole population of your immune cells in adaptive immune response. So now helper T cell, when the T cells will be activated, when the T cell maturation is going on in thymus, as we discussed about the T cell maturation, what did you see there? That there are two selections. One is positive selection, and another thing is negative selection. So if we will revisit the positive selection, then in the positive just a minute, yeah. So in the positive selection, what's happening in thymus? In that positive selection, the T cells which is expressing the TCR, right? In this case, it will express both CD4 and TCR as well as CD4 and CD8 both. So now when this TCR will interact with the antigen which is presented by APC, MHC protein here. These are antigen. This is MHC class 1 and class 2 protein. Class 1 will interact with CD8, class 2 will interact with CD4. So this is your TCR. This is, suppose if it is class 2, then it is CD8, right? So now the CD8 TCR, it will interact with the antigen which is bound or which is presented by this MHC protein in the thymus epithelial cell. Right, so this is antigen. So now this interaction will test, like if the T cell can be able to interact with this epithelial cell which is presenting antigen by MHC. So if it can interact and if the binding is strong, then based on this binding affinity, if it is strong binding affinity, then what will happen? Then this T cells will be selected. And in other hand, the T cells which cannot make this, the TCR of T cell which cannot make this interaction with the MHC, those T cells, see, there is no interaction because this T cell is unable to interact with the MHC and MHC antigen complex. So that's why these T cells will be eliminated. It will be eliminated, and this T cells pool will be selected. But to reduce the autoimmunity, or to reduce the disruption of own cells, self cells, so to make the self tolerance, in the second step, the T cell which will be showing a very strong affinity towards the MHC protein presented by the epithelial cell in the thymus. So those T cells which will be showing very strong interaction, they will be eliminated. Right, that is negative selection. So that's why this negative selection is important, so that it will give rise of T cell which can interact with the self MHC which is presenting antigen. So this interaction should be in a moderate affinity, so then the T cell will be selected. But if the interaction is too much strong, right, then the T cells will be eliminated, because this pool of T cell can actually destroy the own cells, right. So that's why this negative selection is important to give a protection from the autoimmunity, right. Because if you will have this T cell which has very high interaction affinity, binding affinity towards MHC, then it will destroy your own cells. That is called autoimmunity. So that's why this negative selection is important. But if there is any problem in this process, or it can be arise some other reasons also, that we will discuss in later, all the possibilities which can actually give rise of autoimmunity. So but this is one of the possibility, that if negative selection process, this elimination process is defective, then your cell can give rise of a pool of T cell which can actually destroy or they can attack your own cells. So the self tolerance will be compromised there, right. So that will give rise of autoimmunity. So now we will see for some diseases before just going to the mechanism, which are known as the autoimmune diseases. So if you have any doubt in this autoimmunity expression which we discussed, if you have, you can ask. Okay, so now we will discuss about some disease of autoimmune, uh, which is arise due to the autoimmunity. So, which are known as autoimmune disorder. Okay, so we will first start with Hashimoto's thyroiditis. Okay, so here in this two micrograph of no, thyroid gland, if you will properly focus, then left one is the normal thyroid gland, and you can see this is the photo micrograph of a normal thyroid gland, which is showing a follicle line by cuboidal follicular epithelial cells, right? But when you are taking the same sample of, I mean, of thyroid gland, but from a patient who is suffering through a disease, Hashimoto's thyroiditis. So then you can see there is intense lymphocyte infiltration, right? So here the thyroid gland is infiltrated with the lymphocyte. Okay, that is the consequence of your autoimmunity, where your lymphocytes are infiltrated into the thyroid gland, which will give rise of autoimmune disorder that will actually destroy the the thyroid gland. And you will see, if you will see a patient of this disease, they will have a lump in their thyroid gland because of this infiltration of the lymphocytes, right? So in Hashimoto's thyroiditis, which is the most frequently seen in the middle-aged women, they produce autoantibodies against the thyroid hormones, right? Now these thyroid hormones, when they interact with the antibodies, which are actually, those antibodies are generated by the due to the autoimmunity, right? So those antibodies against the thyroid hormone will block them, right? And that will actually interfere the iodine intake, right? And to secrete the thyroid hormone, right? So antibodies are formed. They are against the thyroid protein, which are thyroglobulin and thyroid peroxidase. So both of which are involved in uptake of the iodine, right? So the antibodies will block this thyroglobulin and thyroid peroxidase in the gland. So as they are involved in uptake of iodine, so after blocking of those proteins, the iodine uptake will be interfered, right? So now, as there is lack of the iodine in the gland, then it will lead to decreased production of the thyroid hormone, right? So that is called hypothyroidism. So here, right, hypo means less. So thyroid hormones are not being produced here, or they are producing in a lesser than the normal rate, right? So that is called hypothyroidism. So again, if you will see that in this disease, it will produce autoantibodies. These autoantibodies, what will they do? These autoantibodies will inhibit this thyroid proteins, like thyroglobulin and thyroid peroxidase, which will actually, this blocking of those thyroid proteins will compromise the iodine uptake. So as there is no iodine uptake, then it will lead to decreased production of the thyroid hormone. So thyroid hormone production will be affected. So that is hypothyroidism. Also, it will give rise of sensitized Th1 cells specific for thyroid antigen. So we'll detect the thyroid antigens as an antigen. So now it will so when the sensitized Th1 cell will recognize these thyroid antigens, it will cause the infiltration of the thyroid glands by lymphocytes, macrophage and plasma cell, and all of them will be activated. So plasma cell will secrete antibody, macrophage will show its phagocytic activity, and then it can release some lytic enzymes which can damage the tissue, right? So that's how the cells will be damaged, the tissue will be damaged by the lymphocyte uh, and macrophage and plasma cells infiltration, which is due to the hypersensitization of the Th1 cell, and due to the autoantibody, this autoantibody will inhibit the thyroid protein, so iron uptake will not be occurred, and which will lead to decreased production of the thyroid hormone. So that is called the Hashimoto's thyroiditis, where the thyroid gland is infiltrated with the lymphocyte due to the autoimmunity, right? So the next disease we will see is Goodpasture's syndrome. So in this syndrome, the autoantibodies specific for the for certain basement membrane antigen bind to the basement membrane of the kidney glomeruli and the alveoli of the lungs, right? So when this antibody will bind, they are actually autoantibodies, they are generated by this autoimmunity, so they will bind to the membrane of kidney glomeruli and lungs alveoli. So when they will bind, then as we know that we actually learned from the previous discussion that this, when the antibody recognize antigen, then there is a complement activation, right? So this complement activation will be activated, and that will lead to direct cellular damage, because we saw that when there is membrane attacking complex, MSC forms at the last stage. This MSC will make a pore in the target cell, right? So that's why they can lyse the cell. So that's why this antigen antibody interaction will activate complement, which will induce the cellular damage. It will lyse the cell, and it will ensuing inflammatory response, right? Because of the build-up of complement split product, right? So now, as they will damage the glomeruli and alveoli basal membrane, that will lead to the progressive kidney damage and pulmonary hemorrhage, right? So death can be also caused by several months of developing of these symptoms. So in this micrograph, if you will see that fluorescence anti-IgG, which is staining of a kidney biopsy from a patient with this syndrome, right? So yeah, so here they have used a secondary antibody anti-IgG, which is so now, as we know that in this disease, the kidney basal membranes, they are so they will be bound by the antibodies. So when they use the anti-IgG antibody, which is conjugated with the green fluorescence, so now this secondary antibody will bind to the antibodies, right, which is binding to the basal membrane of the kidney glomeruli. So now you can see those antibodies by the secondary antibody binding. You can see those antibody infiltration by this green color, right? Because the secondary antibody is conjugated with green fluorescence, so it will show the position of the antibody binding. So now you can see that this basemental membrane is rich of autoantibodies, which is generated due to the autoimmunity. So this is a example of autoimmune disorder, which is Goodpasture syndrome. If you have any doubt in these two syndromes, you can ask. this was good pass through syndrome. So after that we will go to IDMM, which is insulin dependent diabetes mellitus. So in this disease, the full form is insulin dependent diabetes mellitus (IDMM). It is caused by an autoimmune attack on the pancreas. These are the photomicrograph of and islets of Langerhans cells, which is known to secrete the insulin hormone. So this tissues from the pancreas, which is an islets of Langerhans, that is collected from normal mouse, and this pancreas is collected from a mouse with the disease, which is insulin dependent diabetes mellitus. So you can see in this photomicrograph, there is infiltration of lymphocyte if you compare with the normal one, right? So again, this is due to the autoimmunity. So how is it happening? Then we can see that the autoimmune attack when autoimmune attack means the infiltration of lymphocyte to this pancreas islets of Langerhans cells, that will destroy the beta cells, because lymphocytes include the macrophage, it include the neutrophil. They are phagocytic cell and they can release the lytic enzymes which can destroy the cell. So that can destroy the beta cell, which will result in the formation of the damaged cells, so which will lead to the decreased production of insulin. And if the insulin production is decreased due to the attack of this lymphocyte, so the beta cells of islet of Langerhans, right? So that will be destroyed, damaged. So now it cannot produce or it cannot secrete the insulin hormone. So due to the lack of insulin hormone secretion, your blood glucose level will be increased, right? So that will give you the diabetes mellitus, right? Now the several factors are important for this destruction of this beta cell. First is the CTL, which is cytotoxic T cell that migrate from the blood vessel to this islets of Langerhans cell, and that will begin to attack the insulin producing cell. They will destroy this cytotoxic T cell, will go and it will destroy those beta cells of islets of Langerhans. That will actually help to decrease the production of insulin, right? And it will secrete the cytokine, for example IFN gamma, TNF alpha, IL-1, right? So that will also activate the macrophage and other lymphocytes, which can damage the pancreas beta cell. Now autoantibody production can also be a contributing factor in the IDM. Why? Because these autoantibodies will bind to the beta cell, and it can facilitate the ADCC, which is antibody dependent cell mediated cytotoxicity. So if they can bind with the beta cell, if they will consider beta cell as their antigen, then it can. Now this antigen antibody complex, as we know from ADCC, that this antigen antibody complex can be bound to NK cell, right? So if they will bind to the NK cell, then the NK cell will destroy the beta cell, right? They will destroy the antigen, that is antibody dependent cell mediated cytotoxicity, ADCC, right? And yeah, so that's how this one possibility is. After this lymphocyte infiltration, the cytotoxic T cell CTL will destroy the beta cell. The other immune cells, for example macrophage, neutrophil, they can also destroy the beta cell. And as there is lymphocyte infiltration, that include the B cell also. B cell will produce the autoantibodies. These autoantibodies will show ADCC when this autoantibody will bind to this beta cell, and this antigen antibody complex can bind to the natural killer cell or NK cell, which will destroy this antigen, right? So that's how this damage of the islet of Langerhans beta cell can be done by this lymphocyte infiltration, right, which will actually leads to reduction of insulin secretion, and it will increase the blood level glucose. So this autoimmune disease is called as insulin dependent diabetes mellitus, IDDM. So now the next is Graves' disease. So Graves' disease, so anyone have any idea about Graves' disease? So the next is Graves' disease. So this is another example of autoimmune disorder. So here in the Graves' disease, so first we will see in the normal case that in normal case the pituitary gland of our brain that secretes the TSH hormone, which is thyroid stimulating hormone, that will bind to the TSH receptor in this thyroid cells, which will produce the thyroid hormone. So this interaction will activate a downstream signaling cascade, which will stimulate the thyroid hormone synthesis and which will be secreted, right? And this regulation is maintained when the secreted thyroid hormones will act as a negative feedback control, right? So it will inhibit this TSH secretion by the pituitary gland when there is excess amount of thyroid hormone. That's how the regulation is maintained, right? But whereas in the Graves' disease, the autoantibodies they are generated due to the autoimmunity. They will bind to the receptor of this here. They bind to the TSH receptor and they mimic the TSH hormone, right? So now, due to the binding of this antibody and the TSH receptor, this interaction will again stimulate the hormone synthesis. So that will secrete the thyroid hormone. Now there is no regulation, right? Because this TSH hormones secretion can be controlled by this negative feedback loop, right? Which is caused by the thyroid hormones itself, right? So by this negative feedback loop. But here, as the antibody is controlling, there is no TSH hormone in the picture. Antibody itself is able to bind to the receptor and it is stimulating the production of the thyroid hormone, right? Now as this interaction is not regulated, that's why it will give rise to the unregulated overproduction of the thyroid hormones. Why? Because in this case, these hormones can control this pituitary gland, so they can regulate the TSH hormone secretion. But here there is no role of TSH hormone, so that's why this regulation is not working here. Whenever this antibody is binding to this receptor, they are producing the thyroid hormone. So that's why it is unregulated overproduction of the thyroid hormone, right? So that will give rise the stimulating autoantibodies, which is Graves' disease. There is unregulated overproduction of the thyroid hormone as there is no regulation. So this condition will give rise the Graves' disease. So there is another Graves' disease, which is myasthenia gravis disease. So this is related to Graves' disease is related to the thyroid cells and this TSH hormone and thyroid receptor interaction. Here this myasthenia gravis disease is related to the skeletal muscle. So for the normal condition, the muscle cell, in order to activate the muscle cell, this muscle cells, skeletal muscle cell receptor, they will receive this acetylcholine from this nerve synapse, right? So this acetylcholine receptor and this acetylcholine receptor in the muscle cell, this interaction is leading to the muscle cell activation. But in the case of myasthenia gravis cell, there are some antibodies which can interact. They are autoantibodies. So these autoantibodies to acetylcholine receptor, so they can interact with the acetylcholine with high binding affinity. So now, as they are interacting with the acetylcholine receptor here, now the acetylcholine cannot bind because the binding sites are blocked by the antibody. So now the acetylcholine cannot bind. So that's why this muscle activation will be inhibited. Why? Because these acetylcholine receptors are saturated, are blocked by this autoantibody. So there is no acetylcholine binding. So absence of this acetylcholine binding, there the muscle activation is inhibited, right? So that's why this anti-autoacetyl, so this antibodies which are against this acetylcholine receptor, which is anti-acetylcholine receptor. This autoantibody will also activate complement, which will damage the muscle cell, because we know from the complement cell activation that at the last stage there is the production of the MAC, right, membrane attack complex. So that will damage, that will create a pore in the membrane, cell membrane, and it will lyse the cell. So that's why they can, so that's why this complement activated complement, they can bind the muscle end plate, they can cause the tissue damage, right? So both this tissue damage will be occurred, and due to the absence of this acetylcholine binding, the muscle activation will be inhibited. So this skeletal muscle, it will become weak and it will become inactivated. So that is myasthenia gravis disease. So now if you want to treat this autoimmune disorder, then first you need to do experiment. So for that you need to experimentally induce the autoimmunity in a normal system. So that is called as experimental autoimmune. So one of the method was taken here, which is experimental autoimmune encephalomyelitis, or EAE, that can be induced in rats by injecting them with myelin basic protein MBP with complete fluid adjuvant or CFA. Adjuvant will help in the antigen activity. So when this normal rat is injected with this myelin basic protein, which is an antigen, and with its adjuvant CFA, then this normal rat will so now this normal rat will face the autoimmunity, right? So now they will be paralyzed, they are EAE rat. So now from this rat we will take the lymph node, and from the lymph node you will get the MBP specific T cell clone, right? Because MBP was the antigen, so whatever T cell we will get, that will be MBP specific T cell clone, that can be generated by culturing the lymph node cells from the EAE. Now this lymph node cells has the T cells which is against this MBP. Now these T cells, which are MBP specific T cell clones, they will be injected to a normal rat, and this normal rat will develop EAE, right, which is autoimmune disorder. So that's how you can experimentally induce autoimmunity, which is encephalomyelitis in the normal system, normal rat. So if you will see the mechanism how this autoimmune response induction works, then you will see there are several ways. Okay, so one by one we will go with that. So now if you will see the mechanism for autoimmune response induction, so here the helper T cell, which when they will be in contact with APC with cross-reacting antigen. APC, they can present the antigen, right, which is a molecular mimicry also. So now this, upon interacting with the APC, which is presenting this antigen, so now this helper T cell will be activated. So it will be uh, activated helper T cell. So now this activated helper T cell, upon addition of this antigen, when they will contact with the APC who are cross-reacting with antigen, they will be activated. Helper T cell will be activated. Then the helper T cell will secrete the IFN gamma. Now this IFN gamma secretion, so now after that, this IFN gamma secretion will help this target tissue epithelium to express the class 2 MHC which will present the antigen, and this antigen is the target tissue antigen only, which is a self antigen. So that will be presented due to the expression of the class 2 MHC by IFN gamma induction, right? So now this helper, so as they can present the antigen, the self antigen, this epithelial tissue antigen, to the helper T cell, now this helper T cell can recognize, right, through that TCR. So as a result, this helper T cell will be activated and it will release IL-2. This IL-2 will in turn activate the cytotoxic T cell. Now this cytotoxic T cell or T cell, that will interact with this target tissue which is expressing the self antigen by class 1 MHC, right? So this CTL will damage this target tissue, so that will cause the tissue damage. So initially IFN gamma will be secreted by the helper T cell which is activated. Then this IFN gamma will be secreted and it will induce the expression of the class 2 MHC in the target tissue epithelium. Now the class 2 MHC in the target tissue epithelium will present the self antigen, which is the epithelial cell only. So it will present the self antigen to the helper T cell, right? Now this helper T cell, when it will contact with this target tissue, now this helper T cell will release IL-2 which will activate the cytotoxic T cell. Now it will be migrated or to this site of tissue, and all of these epithelial cells, they can who are non-APC, they can present the antigen by class 1 MHC. So okay, but in normal case it won't react because in normal case the TH cell would not be activated, would not secrete IL-2, because in the normal case there would not be any production of the class 2 MHC and the antigen presentation by class 2 MHC, because it is a non-APC. So in normal case there should not be any antigen presentation by class 2 MHC. So that's why the helper T cell should not be here. But in this case, as the IFN gamma, as there is overactivated helper T cell, they will secrete IFN gamma, they will allow this epithelial cell to produce the antibody, produce the class 2 MHC to present the antigen. So now it can secrete IL-2 and the CTL will come into the action, and the CTL will bind to the antigen presented by the class 1 MHC, and then it will show its cytotoxic activity. So the target tissue will be damaged, right? This is one pathway or possible mechanism for the autoimmune response induction. Second is this activated T cell can activate the macrophage, and this macrophage can actually do the infiltration of other lymphocytes, and it can actually so basically it is a phagocytic cell, so it can do phagocytosis, it can damage the tissue, and it can release some lytic enzymes which can actually damage the tissue, right? So that will cause the inflammation. So this is another pathway for tissue damage in autoimmunity. Another thing is this activated helper T cell will activate B cell. This B cell will be differentiated into the plasma B cell, which will produce antibody to the self antigen, because this helper T cell that was which is activated because of the self antigen. So the antibody which will be produced, that will be produced to the self antigen, and this antibody which will be produced to the self antigen as a which will bind to the antigen. Now this antibody antigen complex that can invite the complement, it will activate the complement system. So now the complement will come and the complement system will damage the tissue. So that's how the tissue damage can be occurred by these three possible pathways to induce the autoimmune response, which was done here experimentally. If you want to induce, here experimentally, if you want to induce the autoimmunity, then these are the possible mechanism for the autoimmune response induction. If you have any doubt, you can ask. So now we saw the mechanism for autoimmune response induction. So now next is induction of insulin dependent diabetes mellitus, IDDM. Okay, so here if you remember from this IDDM which we saw that the pancreas of a disease patient, the pancreas image was full of lymphocyte infiltration into the beta cell of islets of Langerhans which secretes insulin, right? So yeah, so this autoimmune attack, this lymphocyte infiltration actually destroys the beta cells, which will actually leads to the decreased production of insulin, right? So now if you want to induce this in a normal mice, then how will you do? You can, this is the insulin structural gene, this is the promoter of the insulin. So you can take this promoter PI, which is insulin promoter, and you can fuse to the IFN gamma gene, right? So now this when you will fuse this promoter of insulin PI to the IFN gamma gene, so now this will become a PI IFN gamma trans gene. So this IFN gamma will be actually so this IFN gamma will be expressed under the insulin gene promoter, right? So now and as we know that from this thing that IFN gamma can induce the class 2 MHC. So now the islets of Langerhans beta cell, they have the promoter of insulin. So now if we replace the insulin gene with IFN gamma as a fused transgene, so now under the insulin gene promoter in the beta cell, it will secrete or it will express the IFN gamma, right? It will express the IFN gamma. So now this IFN gamma, as it will be starting expressing in pancreas beta cell, now they will upregulate the class 2 MHC expression, right? Which is very, and as we know that like here epithelial cell is not APC. Similarly, the pancreas beta cell is not APC. So it should not express the class 2 MHC, but due to this IFN gamma expression under the promoter of insulin PI, so this IFN gamma will induce upregulation of class 2 MHC expression. So now the transgenic mice will also express the class 2 MHC by their pancreatic beta cell, right? So now once they, for example here, once they will start expressing class 2 MHC, then the helper T cell will come and they will activate the cytotoxic T cell and other lymphocyte cell, right? So similarly here also, this due to this class 2 MHC expression by the pancreatic beta cell, they will activate the helper T cell here. Then helper T cell will in turn by IL-2, it will activate the cytotoxic T cell and other other lymphocytes, macrophage, B cell, right? So that it will finally lead to develop diabetes because of this infiltration of the lymphocytes in the pancreatic beta cell, right? So that will actually have, develop the tissue damage, and the mice will develop diabetes because the damaged pancreatic tissue cannot secrete the insulin. So blood glucose level will increase and the mice will develop the diabetes, which is associated with the cellular infiltration of the lymphocyte. Here you can see this micrograph, this is the normal one, but this is the, upon the induction you can see that there is a high level of infiltration of the lymphocyte in the pancreatic beta cell, right? So that's why you can, that's how you can make a fuse transgene PI IFN gamma, where IFN gamma will be expressed under promoter of insulin in the beta cell. So beta cell will start expressing IFN gamma, that means beta cell will start expressing class 2 MHC. So it will attract the helper T cell like here, right? And the helper T cell will in turn activate TC cell, it will activate macrophage, it will activate B cell, and there will be lymphocyte infiltration. So all will uh, migrate to this pancreatic beta cell, and they will be activated by the helper T cell like here, and upon the activation they will cause the inflammation, they will damage the tissue, right? So the insulin secretion will be compromised, so the mice will be developing the diabetes, which is known as insulin dependent diabetes mellitus, IDD. So now, if you come back to the treatment portion, how will you treat the autoimmune disorder? There are many ways to treat. So there are certain therapies for the autoimmune disorders are there. For the most of the treatment provide the non-specific suppression of this immune system, right? But the problem is they don't distinguish between a pathologic autoimmune response and a protective immune response, right? Because we need to only suppress the pathogenic autoimmune response, not the normal response. Then your immunity will be compromised. So as the drug cannot distinguish, so it can actually compromise those immunosuppressive drugs can actually compromise to your own immune response, but along with that it can suppress the autoimmune response also. Then after that there was somewhat most selective approach employs cyclosporin A to treat autoimmunity. This agent blocks signal transduction mediated by the T cell receptor. Thus that's why they will inhibit only the antigen activated T cells, right? So it will spare the non-activated T cell, but it will inhibit the only antigen activated T cell. That's how they can be used for the treatment of the autoimmune disorder. And otherwise there was they have developed IL-2 toxin or anti-IL-2 receptor. So the antibody which will bind to the anti-IL-2 receptor, the antibody which will bind to the IL-2 receptor, that's why it will block the IL-2 receptor, so IL-2 cannot come and bind, so it cannot activate the T cell, right? So that's why you can suppress the immune response. And IL-2 toxin that will bind the IL-2 and it will prevent IL-2 to bind here, so that's why also the T cell will not be activated. CD4, as we know that this TCR MHC interaction, TCR and MHC, that is this interaction is stabilized by the CD4. So if we use the anti-CD4 antibody, then the CD4 will not be available, right? It will bind with anti-CD4, so that's why this interaction will be abolished and the T cells will not be activated. We can use anti-MHC antibody which will block MHC to interact with TCR, so that's why it can suppress the immune response, the T cell activation. We can use the peptide blockade. This peptide, which is antigen, which is presented by the antigen, that can be blocked by the peptide blockade. So that can abolish this interaction, and that's why they can actually suppress the T cell activation. And you can use anti-TCR antibody which will block the TCR from interacting with this MHC. So this will also suppress the T cell activation. So that's how you can treat the autoimmune disease by using those suppressors. Right? So any doubt you have, you can ask me. So the next is graft rejection. So we will shift to transplantation process. So when a graft you will transplant to from a donor to a recipient, then there are three kind of rejection can be possible: autograft acceptance, first set rejection, and second set rejection. So the first case autograft acceptance, where you will autograft means when a person is itself donating some of its body part to the same person, right? So if you are if you are donating your body part to yourself, that is autograft. So if your autograft is accepted, that is called autograft acceptance. So here there is example of grafting of epidermis. So this is the grafted epidermis here. So now first day the epidermis will be grafted. Then from day 3 to 7 there is revascularization. So initially due to this after addition of this grafting, so those blood vessels were cut. So after that there will be revascularization. That means the blood vessels will again be formed, right? Day 7 to 10 healing, and day 12 to 14 will be resolution. So that is a good thing that your graft has been accepted. But in case of rejection, there are two types of rejection: first set and second set. In first set, when you graft the epidermis here, and you can see from this color that this grafting epidermis is quite different from the other from the neighborhood. Okay, so that's why it will be rejected. How? So after day 2 3 to so from day 3 to 7 there will be revascularization will be happen. So there will be generation of a new vascular system. Whenever this vascular system will be generated, then the lymphocytes will be infiltrated to the site of this graft, right? This is called cellular infiltration, and then there will be thrombosis and necrosis, right? So the lymphoid cells will actually it will damage the grafted tissue, and there will be tissue the grafted tissue will undergo the necrosis, right? So there will be remnant of the necrotic tissues. So that's why that's how this graft will be rejected here. It is called first set rejection. Second is second set rejection, where again you are grafting the epidermis here, and here in this case, before this revascularization, there will be cellular infiltration, because if it is too much different from your from the recipient, then at the day 3 to 4 the cellular infiltration will begin before the vascularization, right? So as a result, the lymphocytes will be migrated and they will do thrombosis and necrosis. The grafted tissue will undergo necrosis and the tissue will be damaged. So there will be remnant of some necrotic tissue within 5 to 6 days. So the difference is there is no revascularization in the second set, but which is unlike the first set rejection, and it is completed in day 5 to 6, but it takes day 10 to 14 for this completion of the first set rejection. So that is the thing of graft rejection. So now if you want to do a proper so if you want to do a proper grafting, then you should match the HLA or MHC between the donor and the recipient. So if it will be properly matched, then only you can select this donor and recipient as a combo for the transplantation or grafting, right? So for example here, so how this test will be happen? So donor cell and recipient cell will be taken as a microliter plate. So here each of the circle is representing a well. So donor, suppose the donor has HLA allele 2, donor and recipient has HLA allele 1. So they are sharing different allele. So they cannot. So now this donor cannot transplant to the recipient, right? So because they have different allele. So how to know that whether they are different allele? Because if they will donate the graft, this donor cell, to this recipient which has allele 1, so if they will donate, then the problem will be this allele 2 will be taken as a, so yeah, so this graft will be taken as a foreign thing for this recipient, as they have different HLA. So this HLA will take all of their donor cell tissues will be as a foreign material for them. So they will start showing immune response and they will reject the graft, right? So but yeah, so that's why the HLA matching is important for HLA typing. So how will you do that? Suppose donor has HLA A allele 2 and recipient has HLA A allele 1. So now we will add the antibody for HLA allele 2. Okay. So as the donor has HLA allele 2, so it will so upon addition of the antibody to the antibody against HLA A, you will add the complement system. So that, right. So first step is the antibody addition. So antibody will bind to this antigen of HLA. So now complement system we will add. So the as we know that complement system will bind with this antigen antibody complex and it will lyse the cell. So the cell will become leaky. So now if you will add a trypan blue dye or eosin Y dye, so as the cell is leaky, so the cell will take up the dye, right? So you can see the blue color of the trypan blue, right? So in case, yeah, so but in the recipient case, the antibody to HLA allele 2 will not react with the antigen because it has HLA A allele 1. So this antibody will not bind with HLA allele 1. So there is no antibody antigen interaction. So if you will add complement, so there will be no lysis because there is no antigen antibody interaction. So that's why there is no lysis. So now the cell is intact. So the cell, when you will add the dye, the cell will not take the dye. So dye will be excluded. So in this case you will see a blue color. In this case you will see a colorless cell, right? So that means you will conclude that as you have added the HLA2 antibody specific to HLA A allele 2, so that means the donor has HLA allele 2 but the recipient does not have the HLA allele 2, right? So that's how you can type. Similarly, the experiment you can repeat for different antibody to different HLA antigen, HLA2, HLA3, HLA1, HLA4 up to HLA9, right? So that you can test and you can do the same experiment. So for recipient you can see that for recipient HLA antibody to HLA1 and antibody to HLA7, they are showing the blue color, right? That means the recipient has HLA1 and HLA7 allele. The donor 1 also has HLA1 and HLA7 allele, but the donor 2 is showing blue color in antibody specific to HLA2 and antibody specific to HLA3. That means the donor 2 has HLA2 and HLA3, right? So now we will compare the recipient with the donor. The donor 1 is matching because donor 1 and recipient both have the antibody specific to HLA1 and HLA7. So that's why they are sharing the same HLA type. So they are actually matching. Their HLA has been matched, but the donor 2, it has antibody specific to HLA2 and HLA3. So that's why donor 2 will not match. So here the best donor for this recipient will be donor 1, as they are matching with their HLA typing, right? So that is the typing procedure for HLA antigen. Before doing the grafting, you should always do this grafting, HLA typing to match, to check whether which donor is matching with the HLA type with the recipient. That is the purpose of this typing procedure for HLA antigen. right? So this is a typing procedure for HLA antigen. And for graft rejection, there is another mechanism, which is cell-mediated effector mechanism for graft rejection. So suppose this is your graft, so and this is not matching with your, so here suppose the HLA is not matched. If donor 2 will give to donor 1 to recipient, then there will be no HLA matching, so there will be graft rejection. So how it will be happen? This is your graft which is not matched, right? So now what will happen? This graft will be taken as a foreign particle, right, as a foreign antigen. So the immune system will be activated. So first the helper T cell, which is a major controller, that will be activated by this APC. This APC present the antigen to the helper T cell, and the helper T cell will be activated. Now it will secrete IL-2 to activate this DTH, which will secrete IFN gamma and TNF beta. Now this IFN gamma in the macrophage, it will bind and it will induce the expression of the class 2 MHC, and it will activate the macrophage. So the activated macrophage will secrete lytic enzymes which will damage the graft tissue. So similarly IFN gamma in the graft tissue itself, IFN gamma will induce the MHC expression. That's what we discussed. So now the graft, in spite of not being an APC, it will start expressing the class 2 MHC, and it will start presenting the protein, the antigenic peptide through this class 2 MHC because of this IFN gamma induction, right? So that's why it will again attract the lymphocytes, especially helper T cell. Okay, and it will activate the helper T cell, right? Similarly, it will secrete the TNF beta, which will show the cytotoxic effect, and this graft tissue will be damaged. IL-2 will be secreted, which will activate the CD8 plus helper TC cell, cytotoxic T cell. So it will come, it will be activated, and it will bind to the graft tissue, right, which is presenting the antigen with class 1 MHC. So that is recognized by the activated cytotoxic T cell, which is activated by the helper T cell. So now this activated cytotoxic T cell will come, and it will show the cytotoxic effect to the graft, and the graft will be lysed, right? The membrane will be damaged. Then this IL-2 will activate the CD4 plus TC cell, that will come, and that will actually the CD4 plus cytotoxic T cell, they can recognize by, unlike the CD8 plus CDL can recognize the graft with the MHC class 1, but the CD4 plus CTL that can recognize the graft with the class 1 MHC, class 2 MHC. Here class 1 MHC, here class 2 MHC. So now then it will be activated, and this CTL will show its cytotoxic effect to the graft, and the graft will be damaged. Here TH cell will secrete IL-2, 4, 5 and 6, which will activate the B cell. Now the B cell will become plasma B cell, which will secrete antibody, right? The antibody will bind to this graft antigen, and as we know that when this antigen will bind to the antibody will bind to the antigen, then it will activate the complement system, and the complement system will lyse the graft tissue. And also there is ADCC, when this antigen antibody interaction happens, then the NK cell or macrophage, they have the Fc receptor, they bind this antigen antibody complex by this Fc receptor, and they will destroy the corresponding antigen by ADCC effect, right? So that's why these are the cell mediated effector mechanism for graft rejection. That's why these are the ways in which your immune system will reject a graft which is foreign, where whose HLA type is not matched. In this case, the graft will be rejected by all of this mechanism. And there is a example of hyperacute graft rejection in the kidneys. So for example, here if there is any pre-existing antibody, then in the host, so they will be carried to the kidney graft, right, and this antibody will bind to the antigen, to the renal capillary and activate the complement. As you know, antigen antibody complex activate the complement. This complement will damage the cell, damage the tissue, and this complement split products will attract the neutrophils which will release the lytic enzymes, and this lytic enzyme will destroy the endothelial cell here, it will destroy, and the platelets will adhere to the injured tissue here like this, and that will cause a vascular blockage. So that is an example of hyperacute graft rejection, where there is pre-existing host antibody against this graft is already present. That is hyperacute graft rejection. And so, to how to treat if there is any graft rejection, then how to treat this? So the treatment of the graft rejection is you can block the co-stimulator to show energy of the T cell. How? If you remember the T cell and APC interaction, which will activate both the T cell and APC. So this T cell, this APC will present their antigen with MHC class 2 that will be recognized by the TCR as well as the CD4 or CD8, and for class 2 MHC it will be CD4. And there is any co-stimulation signal, this T cell will express CD28 and the APC will express B7. This B7 CD28 interaction is important, that will act as a co-stimulation for this activation, for this T cell activation, right? So now T cells that recognize the graft antigens becomes activated. If they recognize the graft antigen, then the T cell will be activated and the graft will be rejected. Now if you will use the CTLA4 Ig, then this CTLA4 Ig is a inhibitor of the CD28. So now the CD28 will not be available. They will be blocked by the CTLA4 drug, right? So now the CD28 is not available to interact with B7. So this B7 CD28, this CTLA4 is actually, it will bind to B7. So B7 will not be available, B7 will be blocked. So now this B7 CD28 interaction will not be possible because B7 is occupied here, it is blocked by this CTLA4 Ig, right? So now this absence of this co-stimulation signal, there is only one signal, not the co-stimulation signal, but here there are two signals, but here there is only one signal, the co-stimulation signal is absent because of this B7 is blocked, right? So now due to the absence of the signal 2, this co-stimulation signal, the T cell will not be activated, it will become energy. So the immune response will not be created and the graft rejection will be compromised. So the graft will survive. So that's why blocking co-stimulation can cause energy and the graft rejection will not be happening. So that's how you can treat the graft rejection, or you can minimize the graft rejection. Okay. So that's all from today's session. If you have any doubt, you can ask me. Okay, so as there is no doubt, we can actually close the session. See you on tomorrow. See you on the next week.